So the first speaker, it's Dr. Duncan MacGregor from University of Edinburgh. He's a senior postdoc in the Center for Discovery of Brain Sciences. And he will talk us about the role of the hypothalamus in decision-making behavior with some computational modeling. So Duncan, whenever you are ready, off to you.
Okay. Hi all, it's great to be here at Neuromatch, having a brilliant time, all these amazing talks. So this, if you ignore the sort of cumbersome title, is basically about using modeling to predict network structure from single neuron recordings. So basically looking at the patterning single neuron recordings and applying the model, try and fit them and make guesses what the actual network is. So this is work in the VMN and sort of the ventro-medial nucleus of the hypothalamus. Conventionally, it's involved in appetite regulation, but it seems to be involved in various other behavioral things. And we think it's probably a sort of decision-making center. So this picture, this green picture here, is showing you roughly where the ventro-medial nucleus is. This is the third ventricle, and it's the hypothalamus, and you can see these sort of green blobs of the cells. But from this, we're going to get away from the real-looking biology and get into sort of abstract things. So ventro-medial nucleus, regulator of feeding, probably also sexual behavior, also seems to be involved in aggressive behaviors or fight or flight behaviors. It does a lot of things to do with energy homeostasis. So it's involved in glucose homeostasis, thermogenesis, as well as appetite regulation, sends signals to the other parts of the hypothalamus. They're well known at being involved in appetite regulation, make decisions whether to eat or not. It's got various input signals which you'd expect to be involved in this. There's sort of peptide signal ghrelin, which is usually comes from your stomach as empty. There's leptin, that sort of signals your fat reserves, another hormone signal, glucose. Oxytocin, which is another sort of quite well-known peptide signal released in the hypothalamus, is involved in lots of things, oxytocin, but has a big role in appetite regulation. We know it's got output signals that control food intake in various pathways. It also sends controls to places involved in glucose homeostasis. It might send things even to the liver, direct signals. That's kind of vaguely known at the moment. Also energy expenditure things. So the thermogenesis is one of the sort of big ways in which we can make a control over energy expenditure. It's probably a decision-making center, we think, because it's coordinating a lot of different behaviors, which are ones which tend to be exclusive of each other. So you'd have to decide whether to sort of favor one or the other. So it probably is making decisions between these, but certainly it's a center which is doing sort of signal integration and is sending sort of strong signals or involved in deciding which behaviors to activate somewhere. Most of the data we have and understanding what it's doing is from sort of lesioning studies. So if you lesion the VMN, then animals get very sort of obese. Lots of molecular studies looking at the sort of signals and receptors that are there. What we really want to do is try and get a better understanding of how the actual electrical activity, how the neurons are actually processing these signals and what they're doing. So basically we did lots of recording of the neurons. This is work that happened about 10 years ago. I'm in a lab which is primarily experimental. I do modeling there, so really great sort of direct access to lots of electrophysiology data. So basically it's just recording in vivo from lots of VMN neurons. This is single neuron recordings, extracellular in anesthetized animals, and basically process all these recordings by extracting the interspike intervals. So basically you have all your spike times, you count the actual interval between these spike times, and you make histograms of these, and you can process these histograms into these things called hazard functions, and then apply lots of statistical measures of, you're just looking at the firing rate, sort of variation in firing rate, various measures of that, and basically use these sort of statistical measures just to sort of divide these cells based on their patterning into sort of types or potential subpopulations of cells. And you can see here, this is sort of rough idea of all these types, and they're basically just named on sort of descriptive things of all their actual features in their distributions, which I'm going to show you. So if you focus on these, this is the sort of spike rate. This is spike rate binned over 1 second, and this gives you a sort of rough idea of the activity in a cell. It basically just, you can see how noisy the activity is and get a rough idea of the sort of spiking rate. And if you look down, these are different types of cells which are classified. There's not a lot you can see in the sort of spike rate, and you can see where some are faster than others and some are noisier than others. So obviously different one is here is what we call the regular cells, where it's actually very regular spike rate, very sort of regular intervals that don't change very much. This random cell, this is quite sort of typical sort of histogram. You've seen a lot of different types of neurons in the hypothalamus and other parts of the brain. So basically, if you just have random inputs to a cell, a purely random cell will basically give you sort of exponentially decaying distribution. It's basically a sort of Poisson distribution. But most cells have got at least a missing part at the front of this histogram where you have a hyperpolarization that follows a spike, which then gives you a refractory period. So you're basically missing a chunk of the short spike intervals. And then as we go down these different types of cells, we have different sort of features. This, what we originally called a slow DAP, but really turned out to be a bit of a misnomer, but based on other cells that were explained this way, you have a sort of a bulk of very sort of faster intervals at the front of the histogram, and then sort of normal, more sort of random things. And then we have these long tail cells, which has this very particularly long refractory period. So this is 100 milliseconds here, so that's quite, that's particularly long compared to other neurons we've seen. And there are long tail too, it's an even longer refractory period. And then this broad cell, which is a sort of a very wide sort of histogram. And we also apply this analysis called index of dispersion. So index dispersion, each of these bars here measures the variability. So basically this is a sort of way of quantifying noisiness you can see in sort of spike rate here, and we quantify it on different time scales. And this is a really useful way of measuring patterning on longer time scales. So basically, the takeaway from this is your interspike interval histogram gives you sort of short timescale patterning, and your index dispersion range gives you longer timescale patterning. And you can use these to define and then fit the cells. These cells over here, so these ones are variations of sort of random cells at different refractory periods, and nothing too sort of complex and things we often see. These cells over here on the right are sort of stranger ones. So you have the regular cells, and then you have these doublet cells, and these are the doublet ones. Are basically the spiking is made up of very fast doublets and triplets of spikes. So very short intervals, just very, very short bursts. So this big spike at the front histogram is the sort of intervals between these short bursts, and the rest is just the sort of intervals between all these like individual doublets and triplets. And then there's combinations of these. This doublet broad is kind of a mix between sort of what I saw the features of the broad cell and the doublet cell. So it's got a mixture of very sort of long refractory period and also these little short bursts. Then the strangest, most distinct cell is these, what we call oscillatory cells. So they've got multiple peaks within their histogram, and that indicates some sort of underlying rhythm that's driving excitability of these cells. And our idea, which we sort of developed in the model, is that these are some sort of network features are generating a rhythm, and this combined with sort of random input to produce this oscillatory peaks. So integrating FAR-based model is what we've used. I mean, basically it's a very sort of quite simple spiking model, which we then augment by adding sort of hyperpolarizing potentials, these post-spike potentials, and making different combinations of these. So what we call the HAP is a sort of very large magnitude, fast decaying hyperpolarization. The DAP is a depolarization, sort of not as fast decaying, and the AHP is similar to the HAP, but it's a sort of smaller magnitude, but longer lasting. And there's no reset in this version of the sort of integrating FAR model. So basically these post-spike potentials can summate across spikes. And then we can apply these. We used automated fitting using genetic algorithm to basically match the activity in these cells. Basically, you can just take a few parameters, the sort of input rate, the magnitudes and time course parameters, the HAP and DAP, etc. And you can get these really good fits, these really close fits to the spike histograms and also hazard functions. So you can see here, we've got all these quite close matches to these different types of cells. And this then gives us a prediction of what are the intrinsic features that are producing these patterning. And the sort of dominant thing that varies between these is the time course of the HAP. So some of these use quite short HAPs, and some use like longer ones to use, for instance, like the long tail effects, these long refractory periods. And we also have these matches to the index dispersion range, which helps pin down things like the AHP and the DAP. So the other types of cells we couldn't fit with single neuron model. And at this point, the simplest explanations or Occam's razor becomes that it is actually a network feature. And we do know that these cells in VMN, there's not much evidence of inhibitory interconnections, but there is a lot of evidence of excitatory connections in these cells. There's a lot of glutamatergic synapses. So we basically took our fitted integrating FAR model or single neuron model, and we added a very simple model of excitatory synaptic input between these neurons, very simple network. So if we're taking a sort of fit to one of our long tail cells, basically what we're doing as we go down here is we're increasing the density of the synaptic connections. As you increase the density, you start to see the other types of cells emerging from this. So then you move to sort of long tail 1 cell, so this refractory periods become shorter. We keep increasing them still, and this peak, the second peak in the histogram appears, and then we get what's like doublet broad cells. And then if we increase the connection density even more, we start to see basically matches to our sort of doublet cells. And one of the interesting things that get produced in this, if you look at the summed activity over a population of these cells, you start to see these oscillatory sort of patterns. So in a single cell, you have these sort of short, basically these increasingly regular bursts. But looking at population with a bit of noise in the connections, you start to see these almost oscillatory signals. So that was a basically, possible source of the oscillatory signals we saw in the actual oscillatory cells. So we moved on to sort of testing that in another network. One of the other things that came out of this, if we tried, was making a network of different type of cells. So the previous one was just a slow HAP in this neuron. If we add the DAP, which see in other neurons, to this network and tested what it did, we saw this basically bistability. So we had actually seen in the recording some cells that had sort of spontaneous bistability. They would switch between fast and slow firing over quite a long time scale. This example at the top is from the in vivo neuron. And we basically put this in the model, and it dropped out of the very model very easily. There's no explicit bistability in this model. It's purely a sort of emergent effect where we just have the combination of the inhibitory effect of the HAP combined with the excitatory positive feedback of the connections and network, and also the DAP itself. And then we can very simply get a match to this bistable activity, and we can simulate in the model things like inhibitory signals. This CTK is basically a classic gut signal which affects a lot of the hormones, the neurons which are involved in appetite regulation. So we basically just simulated this with a short inhibition to the synaptic input to the model, and this basically drops off and then eventually goes back up to this fast firing state. And we can go further with this by combining excitatory and inhibitory signals. So basically now we've got a network where we can just basically switch on or switch off with a transitory signal, and it will sustain in this high activity state until you give it another intense signal to switch off again. So this is a potential mechanism for decision making. The final interesting network I'm going to show you is how we managed to match the oscillatory neurons. So we couldn't match this with a single cell type network, but we realized we could produce the underlying oscillation that could drive these peaks from a slow HAP network and then feed this into faster HAP cells. So basically we need faster HAP cells so they can get this peak at the front of this histogram. You can't get that if you have a long refractory period. So basically you're using the slow HAP cells to generate an input, an oscillating input signal to another type of cell or fast HAP cells. So the slow HAP ones, their histogram looks like this, and the fast HAP cells are getting this oscillating input, start to see this. We get a very close match to what we'd observed in the recorded cells. And we can kind of see how it's working from looking at the summed activity, the slow HAP ones. We see this sort of oscillating signal. Now, to get this to work and to match the histograms we've seen in the recorded neurons, we had to put noise into the connections to the network. So if you have...
Sorry, Duncan.
Is that time?
1 minute ahead of time, yeah. But,
yeah,
that's the last time, yeah.
Okay, so anyway, basically, slow HAP ones are generating this with a bit of noise in the connections by varying the latency of the connections, the varying delay between the synaptic transmission. Then you get this nice oscillatory waveform, and this produces this oscillatory signal we see in the neurons. And that is that. Thank you for listening.
Thank you, Duncan. Is there any question? There is no questions. Just a quick one. I would like to comment. So there is all these models and external, internal inputs, but how do you associate it to the decision making itself? I I'm not familiar with this type of topic, but where do you translate into decision making?
Well, that's basically, we're basing that. I mean, we think that the VMN is involved in decision making because it seems to arbitrate between these sort of behaviors that can't coexist. Basically, what we think, I mean, we haven't got all the way to an actual decision making mechanism, but we think this bistability where you can switch on and off a signal in a sort of conditional way is, that's a basic mechanism that could be involved in decision making. You've got an all or nothing signal which you can robustly control.
Okay, you don't do actual behavior after these models. Okay, I was just my own bias, I guess.
This is as far as it's got.
Right. Okay, I think we have to move on. Otherwise, we get kicked out of the room. So, thank you, Duncan.